I'm Savannah, and this is my summer research project through UAH. No, that's not good. I'm Savannah, and this is my RCEU project, Kidney Tissue Preparation for 1H-NMR Analysis and Colorimetric Activity Assay of Mitochondria. Metabolomics is the analysis of metabolites in a biological specimen. There are two categories of metabolomics, untargeted and targeted. My project involved targeted metabolomics, where we knew what metabolites we were looking for and we just had to identify and quantify them. The NMR is useful for metabolomics because it's non-destructive and you can easily quantify things like sugars, organic acids, and alcohols. The purpose of my study was to assess the activity of mitochondria and identify and quantify the metabolites present in the kidneys of blue 1WR1 rats. It was important because it helps us understand how diabetes can affect our bodies at the cellular level, and we were trying to decide whether or not the sucrose diet could change the amount of metabolites found in the kidneys, as well as how it affected the activity of the mitochondria. So for my methods, the first part was tissue preparation, and I cut up 180 to 200 milligrams of kidney tissue and suspended it in 6 milliliters of 1:1 hydrochloric acid, potassium phosphate, 100 millimolar pH 7. It was incubated on ice for 30 minutes and vortexed every 5 minutes, and then homogenized with a Dounce homogenizer at 650 RPM with 50 strokes. Then the samples are centrifuged for 15 minutes at 5,000 RPM, and then the supernatants were poured off and lyophilized. To isolate the mitochondria, the pellet was resuspended in 100 millimolar potassium phosphate pH 7.0. Then it was centrifuged at 600 RPM for 30 minutes. The supernatant from that centrifuge step was then divided into 4 Eppendorfs and centrifuged again, and then the supernatants were decanted and the pellets were resuspended in 500 microliters of 100 millimolar K-phos. And then the pellets were combined. This was repeated until there was only 1 Eppendorf left for each sample. The mitochondria was resuspended to a concentration of 0.1 optical density measured at 600 nanometers for the activity assay. So for the actual activity assay, we used Succinate as our reactant, and wells were set up with 20 microliters of 30 millimolar ferrocyanide, 20 microliters of 50 millimolar succinic acid, and 140 microliters of potassium phosphate. 20 microliters of the mitochondria was added to each of the 3 wells. The control wells only had 20 microliters of 30 millimolar ferrocyanide, 20 microliters of 50 millimolar succinate, and 160 of potassium phosphate. The mitochondria were added to separate wells and the plate was preheated for 15 minutes at 37 degrees Celsius. Once the plate was heated, the mitochondria were added and the activity was measured at 30-second intervals for 3 minutes at 420 nanometers. To get our spectrums on the NMR, We used a lyophilized sample and reconstituted it with 500 microliters of potassium phosphate. 125 microliters of 5 millimolar DSS and D2O was added to each sample. The solution was transferred to a 5 millimeter NMR tube, and then the spectrum was obtained using 1 H-NMR pre-sat CPMG with 128 scans and an acquisition time of 2 seconds and relaxation delay of 5 seconds. This is a sample spectrum that was processed using Mestranova that we obtained on the NMR. And you can see the 4 metabolites that we were looking for: alpha glucose, beta glucose, lactic acid, and alanine. The results, we had no significant difference between the mitochondrial activity of the two diets, but you can tell that there was a difference. So the blue is the control diet and the red is the experimental diet, and you can see the statistics below. The results for the metabolites were the same. There were no significant differences between those either. This is for alpha glucose. There's no significant difference for beta glucose either. And no significant difference for alanine or lactic acid. So even though there were no statistical differences found, the mitochondrial activity was lower in the rats on the moderate sucrose diet than the rats on the control diet. So even though it wasn't statistically different, we think that in future studies, if we used a different activity assay or changed some things about our protocol, we might have seen a bigger difference between the two. And then the metabolite concentrations were not statistically different. And these are my references. Thank you for watching.